Will the plastic I'm using for my medical device withstand sterilization? This question is faced by a lot of development teams when choosing a new or even a sustainable plastic for their application. And to answer this question, I brought in Manuel Grandy. Manuel is the co-founder of SteriComp, where they help medical device companies with sterilization and the validation of sterilization. He has over 15 years of experience doing this, and he also works in several standardization groups regarding the topic of sterilization of medical device. In this episode, you're going to learn why sterilization is so important for your medical device success, how to choose the right sterilization method and what the pros and cons are, how to counter material compatibility issues with sterilization methods, and why the compliance to a sterilization standard does not always mean compliance to regulatory requirements. Here's our discussion. Hi, Manuel, and welcome to the podcast.
Hi, Lucas. Thank you.
Yeah. So, Manuel, you're an expert for sterilization of medical device. For those of the listeners who are not very familiar with sterilization, what is sterilization in the context of medical device, and why is it so critical?
Yeah. So, what is it? Usually, it is the last production step after packaging. So, you can imagine even in pallets, you can imagine all your devices packed on pallets. And the goal of this process is to make the product free of microorganisms. And we would ensure a probability of usually 1 to 1 billion. This is the SAL, sterility assurance level, that the device is sterile. So this basically is sterilization. And why is it so important? So there is an obvious clinical reason. So we want to prevent the patient of infections. And how important is this really? So there are some discussions about the infection risk associated with a device, with an unsterile device, versus the risk of infections caused by hospital germs or hospital pathogens. And there's the question, do we really need such a high SAL, or is it really so important that the device is sterile? So I totally understand this discussion. But I'm a sterilization expert, and same applies for notified bodies. So we are not able to judge this, how important this really is clinically. So we come to the next point, why it is important. And this is, it's important for regulatory approval. So often there are delays in certifications or recertifications due to flaws or questions in the validation and the process related to routine sterilization. So regulatory approval is important. And then also there is a big impact on the supply chain. So dependent on the sterilization modality, I have clients, they are waiting like 3 months, and it's not an overestimation, for their product because they're sterilizing with EO. Usually, of course, it gets much faster. And then you can do it in-house or externally. So that is a huge impact on the supply chain. And another important point is the impact on the device design. So dependent on your sterilization modality, you have different impacts on your device design.
Okay, good. So maybe a little more to sterilization. What different methods are there, and what are the pros and cons of these methods?
Yes. So the method of choice is usually the first method is steam always. It's a very old one, maybe the oldest one, and it works pretty well. Only not for heat-sensitive devices, and most of the devices are heat-sensitive. So, for example, in the hospital, if you have metal instruments, they would be usually steam sterilized. But the same applies also in the industry. So in case that you have a cotton, for example, or something that could withstand the high temperature, you use steam. But nevertheless, then in industrial sterilization, mainly EO and gamma is used. So by the way, we are talking, I guess, mainly about industrial sterilization and not about the hospital sterilization, right? So EO and gamma are the two main predominant modalities. And it's like, there are different figures, but like 90% of the overall medical device or sterile medical devices are sterilized by these two modalities. So gamma is very efficient. It works pretty well, but there are often issues with materials. So non-compatibility of materials like Teflon, for example. And also there are some issues with the source, with the gamma. So maybe we can elaborate this a little bit more in detail later. And NEO usually works for most devices pretty well. But the big issue is it's cancerogenic, it's toxic, it's explosive. So pretty much everything, mutagen also. So it's a pretty dangerous chemical, but still we use it because it works so good with so many materials. It has such very good properties of deep penetration into the materials very fast. This is why we are still using it.
Okay, good. Can you explain the two methods, the gamma sterilization and the ETOs, like how it's actually done and what the mechanism is, how it kills microorganisms?
So radiation sterilization, or let's say gamma sterilization, it usually happens in big facilities. They have a cobalt-60 source, so a radiation source. And so you have a big rack of different cobalt-60 pins. And then you have pellets. And they go through, they are passing this source, okay? And you can imagine like 100 pellets around that source moving. And so the radiation, gamma radiation, penetrates pretty well through everything. So around that source, there is like a meter thick or more than a meter thick wall of concrete. So it penetrates easily through medical devices. And what it does is simply it destroys the DNA of the microorganisms. And then they get inactivated. Okay, so this is basically how it works. And then EO is you have a gas, you have a chamber. It's an autoclave, so you can pull down to a vacuum of, for example, 50 millibars. And this chamber is usually between 1 pellet and or 500 liters also, and 30 pellet, 32 pellet. There's pretty big chambers. So you pull down to the vacuum, and then you do some pre-steps, heating and so on also, and of the product. But then the crucial step is you fill the chamber with ethylene oxide. So for example, from 100 millibars to 400. And then this ethylene oxide gets into all the lumen of the device. So first of all, by pressure differences. So from 400 millibars to the 100 millibars of the device, it gets inside, sucked inside. And then there is also a diffusion process. Anyways, eventually the gas will reach to all the important areas, hopefully. And the mode of action is simply, so you have a certain chemical reaction of the ethylene oxide with the DNA again. And this kills again or inactivates again the microorganism.
So talking again about the ETO sterilization, that's basically, so gamma is I irradiate you until you're dead if you're a microorganism, and ETO is basically toxic gas that you use in order to sterilize the product. So your specialty is to validate such a process. First of all, you might elaborate on what validation actually is. So what do you do when you validate a process, and how do you actually validate gamma or ETO sterilization?
So what is validation? Validation is so everything you are not able to verify, you must validate. Okay, so this is a principle in ISO 13485, for example. So sterilization. So what we want to accomplish is this, as I said before, the sterility assurance level of 10 to the minus 6. So a probability of 1 to 1 million that you will have no active or viable microorganism on your device. So you are not able to verify this. You cannot, you can imagine. By testing, this is impossible. So you must, and this is the validation. So you create a certain process, and then you do some testing, some documented testing, of course, to show that the process delivers the results. In this case, the SAL of 1 to the minus 6. So this is the validation, basically, to show this, that your process is able to, on a long term, reach this SAL.
Okay, good. And how would you do that? Do you just like spike it with microorganisms, or how do you walk us through how you would do such a validation?
Yes.
on a top level?
So it depends on the modality, basically. Okay, so there are basically two types of validation, I would say, in sterilization. I mean, there are a few others, but they are like very different modalities. But the two main ones is for gaseous sterilization modalities like EO, VHP, chlorine dioxide, and also for steam that applies. The validation methods are BI based, as you say. Do you spike them? So yes, you do use a BI, and it means that it's a defined number of microorganisms and type also that are used. So usually they are put in a carrier, and this is the BI, the biological indicator. So, for example, it would be 1 million spores of Bacillus atrophius, which we use, for example, for EO sterilization. So you can imagine a strip like this, small strip, with these microorganisms. So for this validation, an important assumption we usually use is these specific homogeneous microorganisms, these types, are reduced log linear over time. So what does it mean? It means that if you have 1 million spores, for example, after 10 minutes, you would reduce to 100,000 spores. Mm-hmm. And after another 10 minutes, if you measure it again, because you can count them, for example, you would measure 10,000 spores. After 10 minutes, you will measure 1,000 spores. So it's a spore log reduction 10 to the 6, 10 to the 5, 10 to the 4, with a constant time, theoretically, okay? This is what we use. And we must show this also, or we showed this also for EO, let's say it like this. Mm-hmm. So this is important. This is the D value. And in validation, you must show that your process can effectively reduce the specific microorganisms in a certain extent. So usually we validate overkill cycles in these gaseous modalities, which means that your sterilization cycle can reduce a BI by 12 logs, mm-hmm. and you adapt the process. So you adapt the exposure accordingly, okay? So dependent on your kill rate and on your D value. So I said before, 10 minutes for 1 log. This cycle would need 120 minutes of effective exposure time of the gas to be an overkill cycle. Mm-hmm. And the easiest way to show this is to show more than, to show 12 spore log reduction and more is this half cycle approach, where you just show a total kill of several BIs. You put into the test cycle, they are totally killed after half of the exposure time. So you showed 6 spore log reductions and more, and then you double it. Then you have this 12. So this is like very basically this basic approach. And now there is, of course, additional important things that you must consider. So in addition to the sterilization cycle parameters, so for example, the gas concentration or the temperature, they have mm-hmm. impact on this 10 minutes, on this D value. The higher the temperature, the shorter this D value. So you make it instead of 10 minutes and 5 minutes and so on. This is clear. You must consider this, but this you keep constant for your cycle. But now there is another important factor in validation. So for example, the density of the load is also important. You also consider this in validation. And another crucial one is the design of the device itself. Okay. So for example, if you have long hoses, or for example, places as between 2 lips of the rubber of a syringe, they are of course far more difficult for the gas that I explained before, this EO gas, for example, to reach, okay, than compared to if you put the BI outside that syringe. So that kill rate I described before, that D value must be somehow shown at these locations. Mm-hmm. Otherwise, it does not make much sense, okay? And that's why these most difficult to sterilize locations of the device are so important. We call them like this. They are so important for the significance of your validation. There is currently a lot of discussions around that. So I thought it backwards and forward, and I personally see no other reliable way than placing the BI at these locations, at least as a general approach that would be described in the standards. But maybe also a topic that we can elaborate more later. This is more or less the basics in gaseous sterilization.
Oh, okay, good. So to summarize for our listeners, so what you do is you can never measure 1 in a million, and that would be verification of the sterility. And because you cannot do this, the only thing you can do is try to understand the system, try to understand the mechanics and so-called kinetics of your sterilization. So I know if I have 1 million germs, that like 90% will be killed in 10 minutes, and then also 90% in the next 10 minutes. So I know it's an exponential curve, and if I can show that this exponential curve exists, I can say I will reach the sterilization assurance level of 1 to 1 million in this amount of time, then I double the time, and then I'm really, really sure that I am sterile to the requirements. And then it also depends on where the germs will be placed. So if they're placed on a very, very, I'd say, difficult to reach location, they will survive longer. If they're just on the surface, they will survive less long. And you say, just assume the worst case for the placement of this biological indicator. Is that what you're saying, basically?
That's correct. Because if you don't do this, then there will always be the question, well, what about this location? What is the d-value? What happens if there are 10, 15, 20 germs?
If you are a medical device company and you're looking to make your devices more sustainable, there's a link in the description for you. Okay, you mentioned that you're in several standardization groups and also very active. And you also teased that there's a discussion going on where to put the biological indicator. Could you elaborate on that? And does that mean that standardization, how is the conflict between standardization groups and regulatory requirements? Do they always match, or how is that?
I mean, basically, what is in the standards is the regulatory requirement. But the standards, I must say, they are more and more moving away from something that you can scientifically explain. And I see this in different instances. And one is this placing of the BI discussion. Okay. So you can imagine, as a manufacturer, it's a pain. You do exactly what is described in the standard, for example, a method. And I can elaborate a little bit more about this on this regarding the BI placement. And you do it, you do your whole validation on it, you do your product test on it, you send it to a notified body or authority, and the authority says, wait a second, I don't understand this. Please explain it to me. Well, it's written in the standard. Yeah, but still... I have a question. Can you explain this to me? It does scientifically not add up. And then you have a very big conflict. Okay. It's a pain for the manufacturer. Maybe they must start from scratch again, their product testing and so on, at least their sterilization validation and so on. So that could be really a serious issue. So the job of the standard is to make sure that you have patient safety and that it's based on scientific knowledge, what we can accept and what a notified body can also accept with a scientific background. And there I see quite a few instances where this is unfortunately moving away a little bit from good practice in the standard work. And one discussion is about this most difficult to sterilize location. So there is a practice that is quite practical for the test. So I explained before that you would place the BI at the most difficult to sterilize location. Yes. Why this is important and so on. So there is an approach where you would say, do not place the BI at all in the product. Leave it out. Use some kind of PCD, for example, let's say a 1 meter hose, which is a standard PCD, and compare this PCD with your product bioburden in a sublethal cycle. So it's a very short cycle, and you will have growth on your PCD, where the BI is placed.
What's a PCD?
It's a process challenge device, a hose, for example, which is a challenge for the process, and the BI is placed there, this bioindicator. So the question is, well, how do I relate this PCD, which is some very different thing, to my product, to my syringe with the double lips, for example? How do I relate this to the product? And then this questionable idea is that you compare it in this short cycle and you show growth on the PCD and no growth in the test of sterility of, for example, the syringe, let's say. And then you say, well, this syringe is easier now to sterilize. So let's forget about the syringe, and now we do everything. So the process development and the validation with this PCD, because you showed that experience. And you compared it. It sounds logical, but if you have a closer look on it, then there is a simple question for a notified body, for example. Well, how did you show that there was, let's say, enough lethal conditions in the syringe to reach the SAL of 10 to the minus 6? How did you show this? 12 spore lock reductions. Because we do not know if there was microorganisms in this syringe, inside of it. And if there were microorganisms, we don't know what kind and so on. So you're missing a very important information, this d-value, in your product. And that's why you cannot explain to a notified body what is your SAL there. So a very simple question. You follow the standard eventually in case that it goes live like this, and you will face issues.
First question there. I try to summarize. You have a hose that you use as process challenge device, and then you have a syringe and you place into both a biological indicator. You run it through your sublethal sterilization test. For one, it will lead to no growth. So the syringe has no growth, and then the process challenge device, which is a hose or whatever, has growth. And then you basically show that the syringe is less difficult to sterilize than the hose, which is no wonder. And then you do everything, the validation on your process challenge device, and forget about the syringe. And then the question that you cannot answer is that how do you know that you will get to 10 to the minus 6 on the syringe? And when do you get there? Is that the correct summary?
Yes, exactly.
Okay, good. And now I'm a development team of a medical device, and I have a new medical device. I want to make it, for example, like the usual case, they work with me. They try to make sustainable plastics or sustainable medical devices, and I have no clue about sterilization. How do I choose my sterilization method? What are the pros and the cons? And when would I be, let's say, involving someone like you?
So how do you choose the sterilization method? I would usually start with the 3 methods I told you already. This is steam, EO, and gamma, or maybe E-beam. So these 3 methods they have a lot of advantages. This, for example, that there exist many companies providing such services. So that would be the first. Then there are, and I guess this could be also something we elaborate in more detail, there are alternatives that sometimes make sense. But you would start with these 3 or 4. And yeah, so there are material compatibility is one very important aspect. So depending on your device, you will have certain materials. So very simple, an instrument of stainless steel is an instrument of stainless steel because it cuts very well. So you will probably not change that to another material, just that you are forced to use EO. You would just use steam in this case. But for some devices, you have certain materials and you would adapt the validation method accordingly. And this is a quite straightforward process, let's say. So you have your device, you have an idea what materials you use. That will be maybe the first step. And then you look at the very early beginning of the development process. You look what could be the sterilization method for this. There is for example, document, the AMI-TR 17. It is a very good source that is telling you for these kind of materials what could be the right sterilization modality. And then if you're starting to question it, then you must dig deeper. If you say, oh, wait a second, this material doesn't work with the sterilization modalities we would primarily choose. So then maybe you would think about to change the material and to adapt something in the design or so. You say, well, I want to sterilize it. I have these materials, so EO is the method of choice. Then there are certain other aspects like all areas must be accessible for the gas to penetrate. So you would have influences on the device design also, maybe.
Okay. So maybe my 5 cents. I say irradiation sterilization has a lot of advantages. You have a very deep penetration depth, so you can use pellets. It penetrates quite a bit, especially gamma. And the big disadvantage of this is that it causes defects and so-called radicals in the polymers, which will, when not properly stabilized, so when you do not use the proper additives, will lead to a degradation over time, and that is called aging. So what happens, for example, with the polypropylene, you shoot it with gamma, it's not stabilized. You will throw out electrons of the hulls of the atoms, and then first it will be fine, and then after 2 years, or half a year or a few months, it will start to get brittle because there's an auto-oxidative process going on, which basically degrades the chains. And that is one big disadvantage. So like if you want to use irradiation sterilization, you need to use the proper material and then it works. Then with ETO, you're much more constrained with the device design. So you need to have open lumens. If you can't have that, you need to have a material that is very, let's say, able to diffuse the ETO through it. For example, polyethylene is very nicely able to let the ETO through. That's also the reason why usually in medical packaging we have a PET blister and then a Tyvek, which is an HDPE textile on top. And that lets the ETO through nicely. That's the reason why we use these blisters, because they are they allow the diffusion of ETO. So that's my 5 cents on how to choose the sterilization method. Usually it's very funny because your perspective is, yeah, we have this material and we have this device and now we need to sterilize it. And I'm like, yeah, we usually want to go for a sterilization method. So I choose the design and the material appropriately, because like I'm much more on the design side and you're much more on the, okay, we have this device and now we choose the right sterilization. And then the environmental impact, what would you say?
So it's a contra for both. They have issues, or let's say a bad reputation with regards to that. So gamma sterilization, this cobalt 60, it's quite an issue because also it's not very environmentally friendly to produce it. It has a very good and excellent CO2 footprint. So this is a big advantage. EO has worse CO2 footprint. So it actually needs a lot of energy. So it's interesting. I know a provider in a not so small city, and they are by far consuming the most electricity. And then there is also this issue with the cancerogenity. So the, let's say, pollution of the environment, which you can't control with filters and so on.
Okay. So what would you say is like ETO is much more the pollution of the surrounding area with the toxic gas, or the CO2 footprint, the energy usage?
I would say it's the CO2 footprint because, I mean, if you look on the facts, because the surrounding of the area, if you have good filters, and we do have good filters in Europe, and we did since a longer time, then I don't think that there is an issue for this surrounding area.
Yeah,
but so it's a hot topic. It's a hot topic. So if you know that your kids are playing next to this plant, then a lot of experts can tell you that this is no problem. You will have some thoughts about it.
Yeah, sure. I think also, did I misremember that the FDA is looking into this? It's like cancer rates around ETO sterilization units, and they are a little higher than average. Is that right?
Yeah, this was the EPA, so another agency in the US. And indeed, there were some statistics and so on, and there was a huge topic around it in the US. With hearings, you can watch on LinkedIn, on YouTube, about 3 days of material. People in the areas that got cancer, and they tell their story, which is heartbreaking, of course. So yes, there was a big topic. Was the big topic in the US? Yeah, the question is really, when did this pollution happen? Did it happen in the 90s? Did it happen before they installed filters? And this is maybe something where others that looked into it into more details, they say, well, is this still relevant for now? Is this relevant if you install proper filters?
I think like this is a little philosophical, the discussion now, because like if you pose the question is what is doing more harm, the sterilization method? Because if you can, like if there is the possibility of having pollution and people getting cancer, for medical devices that should be safe and effective and helping patients. So there's like, as always in the medical device field, you have to make a risk assessment. The benefits have to outweigh the risks. So what do you think? Is it still justified to have ETO sterilization?
Yeah, I am expert in EO sterilization mainly. So since many years, I do look into the other modalities, and it's also from business-wise very interesting for me. So the next few years. So, but I'm not 100% unbiased, I must say, but I try to be unbiased. So I absolutely believe that we will have EO the next 10 years for sure, and maybe even longer, I believe, because it has quite a lot of advantages, and I think it's manageable. So if we have tomorrow an alternative that works as well as EO, for a wide range of materials and different designs and so on. Let's use it. Let's get rid of it tomorrow. But there is this is not on the radar right now, I would say. And on the other hand, I mean, we can manage such substances. We do it in the industry. We are still driving cars that have CO2, but also have cancerogenic substances. We use filters to manage it. We use other chemicals in the industry we cannot get rid of, like formaldehyde or so. This will also stay, and it's manageable, I would say.
Yeah, I think it has a lot of analogies to I'd say the fluoropolymer use, because like fluoropolymers are in a heavy discussion also under the PFAS ban of the ECHA. And the point is or the best argument I've found against fluoropolymers, because if you use fluoropolymers in your medical application, there is strong evidence that fluoropolymers are safe, that they do not cause any adverse effects, and the only thing they have against them is that they're persistent. But that just means that they're very chemically resistant, which is something you want for an implant. Because if you have it in your body, you don't want it to dissolve into other chemicals. It's a stupid idea. So you want something that is very chemical resistant, low leachables, and high biocompatibility. They can do that. The best argument you find against the fluoropolymers, and there they have a point, is during the production, there has been in the past a lot of contamination of surrounding areas. And you need to make sure that your production facility is clean, doesn't leak into the environment. But the societal benefit of using fluoropolymers, it's certainly there. We use it for minimally invasive surgery. And there, certainly the benefits outweigh the risks. I don't say that we necessarily need Teflon pans. I don't know if we need that. I like iron skillets. And it has an analogy. So do you have an alternative? What is the benefit and what is the risk? And we should have this benefit-risk approach. Maybe you could also elaborate on what alternatives we would be using for ETO sterilization.
Yes, so for EO, there are there are a few methods in the pipeline. There is also, we meet together with, we just met in Munich a month ago with around 120 experts from the whole world to discuss on these alternatives. So there is a lot going on and we work a lot on this. So the main candidates are ClO2, NO2, and VHP. These three are the main candidates. I would say, did I forget one? Well, and then there are a few others also in the pipeline that could work. So the good thing is that they are far less harmful, of course. Otherwise, it would not make much sense. But they also have quite some disadvantage. So the main disadvantage is, or the main advantage of EO compared to these methods is that it can simply penetrate better into the materials or into the product through pellets. So you use a pellet with a thick cardboard or several cardboard layers. You use a long lumen and so on, and it penetrates inside. And for these alternatives, you would usually get rid of the cardboard. Not always. It could work also with the cardboard. But first of all, you would get rid of this cardboard, which makes it much more difficult for a sterilization service provider, for example. So you would go then in-house. And then there is the material compatibility. So all these three candidates, they have one big difference to EO with regards to mode of action. They are working by oxidation. Okay, it's oxidation of the DNA, and this is much less specific. So it would also oxidize the materials or react with materials, so that regarding material compatibility, and this is the second advantage of EO, they are weaker. Okay, so they work perfectly with some materials, but they're then very bad with others. And this is also something you can find in AMI TAR 17. But nevertheless, I'm very interested in these alternatives and to bring it to the client. What mainly I think is very interesting is that it's a good reason to go in-house.
Yes.
So look at steam sterilization versus EO. Steam sterilization, 90% do it in-house. Why? Because it's steam only. But with EO, it's the other way around. 90% send it to some other company because they don't want to have this chemical in-house. It makes a lot of sense. So with these alternatives, you could again say, well, I have a modality that works. So I can very smoothly bring this into my production in line, let's say, inside of in my clean room, pack it after the sterilization directly in my facility, and then send it out to the customer. So that's for me, one thing that is extremely interesting when looking to this alternatives to EO.
So the alternatives to EO do not only unlock or mitigate the environmental impact of ETO, but they are also enabling, due to being less toxic, to bring that in-house, because no one wants really like a toxic gas station in your own house. So that is an advantage. Can you comment on how many medical device companies, let's say in Europe, have an own ETO sterilization, and maybe also who has a gamma sterilization in-house?
It's good that you asked this, because you said nobody wants this. That's not true. There are quite a few that do it. I would say like in Germany alone, like 30 companies
have an ETO.
There are also there are also some very small sterilizers. And, it is manageable. So you can do it, but you have to, it's always a project with some risks involved, also regulatory risks. It can be very complex with the authorities. If they hear EO, they read a little bit and Google a little bit, for example, and it can take years to get this approved. Mm-hmm. And it can take you some super extra measures and so on. It's a difficult project, but you definitely, if you know what you're doing, you can do it. It has a lot of advantages, and you can do EO sterilization in-house still. In Germany, at least. In some other countries, it's far more difficult. And gamma is very difficult in-house. So I only know one company in Germany doing this.
Yes.
Because, bringing the new pens alone is a castor transportation. So it's very complicated, very expensive. And I guess for the approval, you would also need like 10 years or so, I could imagine. So it's very complex. So you would usually do externally.
Yeah. It's only a very big one who has a gamma sterilization in Europe, all of Europe, I think, because as you said, you need at least meters thick concrete walls. Like yes. building the place takes you years, and then have it validated and all of that. It's a huge effort. It's a huge investment. You only do that if you do a lot of product. And yes, they are doing a lot of product.
But there is talking about in-house and radiation. So there is an approach it's called low energy radiation. So if you reduce the energy, the megavolts, you don't need meters thick wall. You can reduce it to, I don't know, for example, 40 centimeters or so, or half meter or so. And then it gets interesting to do this in-house with lower energy and with not with an electrical source of radiation.
Okay, good. So maybe a little bit about your company, SteriComp. How would the company get in, or how and at what stage would a company or a development team come into contact with you, and what do you help them with?
So they come into contact very early sometimes. So just now we started a new project with a startup. They already did some first clinical trials, and then they want to go on the market in 2 or 3 years. So they are maybe later. So they are now starting to understand, let's start a sterilization validation. Let's validate the process and start the CE certification. So this is one of the cases, but some of them come also very late. For example, when they already wanted to approve their sterilization process, and then they get questions from the notified body or authorities, and they ask, what can we do now? So often difficult situations, and they come also at this stage of the process. Well, or also at the very early beginning, when they understand what could be our materials, what could be our related to our sterilization process. Mm-hmm. We also have this kind of projects.
Okay. Yeah, I mean, that is something I always discover is some people only learn with pain. So if they have the notified body asking questions, then they'll come to you, not before that.
And maybe you would not expect that,
Yes.
that it's so difficult or that it would face these difficulties.
Yes. Sometimes people think it's going to go well, and then you get questions, and then they come to you, and then you need to help them firefight. Yeah, it's always the same with material selection. It's very similar. Some you get involved at a very early stage, and some you get involved when you have either complaints or failed CE marking attempts, and then they come to you and say, hey, we have this material, it doesn't work, what can we do, what can we take, because we need a solution basically yesterday. And that is very typical. Good. So
this is the validation part. So we also have the equipment part. So most of us are engineers in the company. So we started with the equipment of sterilization, and still we provide then consulting in what process to choose and what equipment to choose also, and how to implement it in the production.
Yeah, I mean, that is a huge part of it. What exact equipment, what process, and how do I get the process validated? That's a huge part of it. So Manuel, what advice would you like to give to our listeners in regards to sterilization of medical device?
What is crucial in sterilization, I think, is that you have somebody in the company that has a basic understanding of sterilization, and so subject matter expert. And there are also somebody from quality who gets into it, and there are great platforms also to get into it. So SFSAP, it's the Society for Sterilization Assurance Professionals, for example. Their mission is to improve the knowledge in sterilization and the science behind it. Another important aspect, I think, is that the regulatory approval is a very big and important part in sterilization. It's often a showstopper in CE marking. So a good relation to your notified body and have an exchange to your notified body. And this is now possible with, you can have a dialogue. There was a change in the MDR or so a few years ago. So you can have a dialogue with the notified body or maybe also with your authority. This is also, I think, important to understand exactly, sometimes to understand exactly what is requested, let's say.
Yeah, that is certainly the case. The structured dialogue with the notified body is important. So maybe a few last words from you.
Actually, nothing to add.
Okay.
Thank you very much for the interesting conversation.
Yes, thank you very much too. Have a very good one.